I'm Wendy Hartsock, science and peptide enthusiast. On this episode of Exploration Science, we dive into some of the intricacies of personalized medicine. I sat down at the 2021 TIDES meeting in Boston with Trishul Shah, the Director of Business Development, as well as the Head of North American Sales for PolyPeptide Group, and Jon Collins, the Vice President of Business Development for CEM. We discussed some of the challenges as well as the opportunities for peptide-based personalized medicine.
So thank you both for joining me for this.
Thanks for having us.
Yeah, absolutely. So Trishul, I know you were here, you were teaching a workshop. You had a whole workshop that you actually were hosting, and then you had a specific session in that.
Yeah.
What was your session?
So the workshop was focused on the new ANDA guidance for peptides,
Okay.
from rDNA origin. So we had 4 speakers, and I was one of the speakers. We had Jay Zhang from the FDA, Annie de Groot from Epivax, and Garrett Haas from Baucom. So it was a good discussion on the new guidance and how we approach that.
Okay, excellent. Yeah, that's great. I think, some of the people that I've talked to just in general in the industry, there's a lack of information out there on preparing IND packages, on just basically how do you get from a discovery compound into a human, that whole process, right? So,
Yeah.
so excellent work on organizing that and running it. Yeah, I wish I could have come. Something I really wanted to talk to you about, and Jon as well, is personalized medicine. I think this is something that we had heard about years and years ago, right? People kept talking about it. Oh, it's going to be personalized vaccines. We're going to have individualized medicine just for one specific person. But there was a lot of people out there who was like, no, that's not going to happen. It's too hard, right? So maybe you could talk a little bit about what's the current scientific state of personalized medicine, and then also what is the market for it and where is it going?
So personalized medicine has been quite a phenomenal. Pre-pandemic, it was taking off. There have been a number of companies, mainly in the Boston area, focused on personalized medicine in different sectors. So you had the CAR-Ts, like with the Kites and the Gileads of the world. And then it sort of ventured into peptides being used together with checkpoint inhibitors. And that was a whole new area for a CMO like us, because the traditional peptide therapeutics, when you manufacture them, it takes 4 to 5 months. You go through a full process development, and all of a sudden now you're making 20 to 60 peptides in a matter of weeks and under GMP. So that was a whole game-changing, mind-boggling concept that we had to come around to and understand and discover and find a pragmatic way to actually manufacture that. And we started looking for partners, and this is when we met with Jon and CEM and how those instruments could be used to actually produce those peptides very quickly. So through the pandemic, the market has sort of slowed down due to the focus on COVID vaccines, but again, the same concept can be used for COVID vaccines, and we saw what happened with Moderna with Pfizer and how quickly they came out with their vaccines, and then J&J. So a similar approach can be used for cancer vaccines, and then I think now with the mRNA frontier, that brings a whole new aspect to this whole arena of immunotherapy.
Right, yeah, absolutely. So you brought up CEM and technology. How is that impacting personalized medicine? I mean, you sort of alluded to it, that it's getting us there, but what is it about the technology? And then Jon, maybe you can chime in on sort of where it's going and I guess where the foothold is for CEM in this.
Absolutely. So I think one of the great things about the CEM instruments and synthesizers that CEM manufactures is it allows really rapid manufacturing under heating to obtain the accrued peptides, but then also having the ability to sort of cleave and then purify them. I think one of the challenges that we faced, of course, is although we've been meeting the needs, is the bottleneck is now in the purification, and getting to a place where we have automated purification, automated systems would take us to the next level. It would help patients because some of these patients are critical and they need the peptides in that short time frame without any delay. Otherwise, it's too late.
Right. And the quantity of peptide, we've talked about this before, the quantity of each peptide, is it in the range of 30 to 40 milligrams? Is that what?
Yeah, typically it's in that 20 to 50 milligrams, depending on the program. And and the plans by the sponsor what they're going to use them for. There are some programs that are more long-term where criticality and timelines are not that important. So because then you start looking at formulation and stability studies. However, in some cases they are critical, and so the smaller amount of peptide is usually useful.
Right. Okay. What are the salt forms that are used in these?
Most of the time it's TFA, but sometimes a peptide is not conducive to a TFA salt form. So we would obviously explore other alternatives like sodium or ammonium or chloride.
Synthesis obviously is a big deal because you need to have a lot of peptide at one time you're going to get the peptide that you programmed into the instrument, that's what it's going to put out. But purification has always historically been the biggest pain point of the peptide chemist, right? And so it'd be interesting to start thinking about, especially helping with salt forms, what can companies do to help that process as well?
Absolutely. Yeah, it's been, I think as Trishul was saying, the challenge for this whole field of needle to needle, where you're taking a sample from a patient and then when you can actually inject a drug back into them, trying to make that as quick as possible. So it's been really interesting working with PolyPeptide to see the whole picture holistically. How can we help? And we've taken that challenge. So it's historically been on the microwave side for the synthesis to get your peptides as fast as possible, but also to try to really use it so that you don't have to do resynthesis either. You get high purity each time you can make it with the microwave. But then really also we've been looking at the purification process, with our new system, the Prodigy, using elevated temperature to try to really bring that in line for more fast purification that also fits the scale range, where you can purify hundreds of milligrams really fast, low amounts of solvent, and get really good separation the first time too. So you bring the whole process together to try to make it as fast as possible. We've tried to streamline the cleavage process as well with an elevated temperature system. But then, as you mentioned, also considering to even look at the lyophilization, salt forms, bring that whole step together to try to bring that together.
Yeah. In general, there's obviously challenges. Just cost, I've heard, is like probably one of the higher thresholds of challenge of like, what does that look like for the future? Who can really afford personalized medicine? Right. How do you get the cost down, and how does technology help?
Yeah, that's a really good question, and I think secondary to that is insurance payout and reimbursement. How does that fit into all this? You're trying to get these peptides to patients quickly, needle to needle, and the process itself is not that expensive relative to therapeutic peptides, but it is expensive for a single patient. When you take into account the sequencing, the formulation aspects of it, and then actually the administration aspects of it, it is expensive. And I think the way to get it down is get a point of automation, and that will help bring the cost of making the actual peptides. But you still have other areas of pain points. The formulation, the sequencing, all that's very expensive as well. So across the spectrum, I think you'll get to a point where there is a limit, but.
So actually, a couple of questions around that. Maybe we could just take a step back. Can we just go through the whole process, needle to needle?
Yeah.
Can you just give us an overview of what that process is?
Yeah. So generally, what happens is a patient that has cancer, they'll get a biopsy. After biopsy, the cells are sent over for sequencing, and they obtain all neoantigen peptides, typically in that sequence of the cancer tumor cells. You have the antigens and the neoantigens, so the neoantigens are identified. Those sequences are then predicted. So that's the key difference between a lot of these companies is the prediction model. They will identify which ones they believe will be successful, and those are sent to the CMO, like ourselves, to manufacture. So now you get to another threshold where some of these are difficult to manufacture, some are relatively easy. Murphy's Law, the difficult ones are the ones that you really want. So it's finding that nice balance, and then it gets switched over to the formulator to formulate it. And there's a part of that is also there might be pooling of certain peptides.
Right. So that was kind of, okay, let's finish the process. And then, because that was my next question, when you start mixing peptides, bad things can happen. But So first off, so you get to that point where you formulated and then pooled and then straight to patient, or?
Straight to patient.
Okay.
So in a lot of cases in the early stage, you've got pharmacies or hospitals doing the formulation bedside.
Wow. Okay.
Yeah.
Huh.
But, as you said, you're mixing multiple peptides, so we talked about salt forms and residual solvents. How do you control those? And speaking with the FDA, you do have to do some sort of testing just to prove that these are at low levels. But in general, the safety risk versus the risk of the patient needing it has to be balanced as well.
Right. So then that's kind of another avenue, right? Is like, okay, so yes, these are good for oncology patients.
Yeah.
Where else does personalized medicine fit in? I mean, you had brought up COVID, but there's got to be many applications where if it were affordable, it would make sense.
Yeah, and that is interesting because with oncology and patients that are at that late stage, I think you can justify it. For more normal diseases or traditional diseases, it might become, it's a bit of a challenge because then you do have to probably characterize the impurities, the solvents, the counterions.
Right, yeah.
So, and I think it's a good discussion which the FDA is still trying to get a handle on and working through.
Okay, and yeah, so is the FDA very involved with the whole community and
Absolutely. It's
communicative?
Yes, the CBER division of the US FDA, and honestly, they've taken the lead in the world. So you see a lot of that coming out here and out of fruition. I do know that PMDA is also looking into this now in Japan, the European authorities also, but the lead is really followed by the US FDA.
Do you see any pooling of even to like subgroups of multiple patients that could take one batch? Is there like common mutations that could maybe help on the economies?
So that's a new area that they're looking at, which are like these shared antigens. So what they will do, so it's sort of like a balance between the traditional warehouse approach and this personalized approach where you can make enough of the large amounts of these peptides, of these new antigens, and you could then share them between different patients. So that's, again, some companies are looking into that and they're seeing that. There have been companies that have said that if I have a new antigen and you have the same new antigen, it might be a new antigen for me, but it could be suppressive for you. So you could have the opposite effect for you. So there's that discussion as well, and that's where the prediction models become important.
Yep.
So I think it's still a field that's in its infancy and still growing. There's a lot coming out, a lot of knowledge.
Right. And you're targeting both the CD4 and the CD8 response, right, with some of these new antigens?
Absolutely, yeah.
So do you think that there is one better than the other, also personalized? Like maybe John's CD4 response would be better for him and my CD8 response would be better for me? Or like where does that... Because I know like you talk about suppressive peptides, but what also just about engaging the systems and the types of cancers? Like, are people looking at that?
So yes, there are companies that are looking at the different ones. There are some companies that prefer just the CD4 versus CD8. Some that are looking at both or a specific ratio. And I think it also depends on the cancer type and the location of the cancer. So they're still trying to work that out. And that is sort of the technology behind it that differentiates one company to the other company.
Yeah. And then along those lines, so we're making these mixtures of peptides. Could you also just make one peptide that is has specific cleavage sites that would very quickly be cleaved into those peptides that go into the antigen presenting cells? Is there a reason why we can't just make one long peptide that has multiple neoantigens?
Yeah, I mean that's a good question. So I guess the problem with that is when you make, and as you know, as a peptide chemist, as you make longer peptides, the more impurities you get, the lower the yield.
Yeah.
And that's where the challenges start. And so there's also a concept of how long should the sequences be, and how much is there overlap between sequences of the protein that's been looked at. So some of these companies are also looking at ratios of should they have long peptides and how long, should they have short peptides and how short.
Right.
So it's a mixture between the two.
Yeah, okay. What's the right answer? No.
We typically hear, I guess, between 15 and kind of 30 amino acids. Is that what you're seeing for
Absolutely,
typical neoantigens?
Yeah, absolutely. That seems to be the range.
Yeah.
And there's some which I'm seeing shorter, but again, it depends on the cancer. the technology behind the company that's coming out with these sequences.
Is there any certain types of cancer that you think this is going to be the most promising for, or?
Not really. I mean, I think a lot of what we've seen has been in melanoma or bladder cancer, but I think there are companies that are looking at different areas in glioblastoma as well. So.
Yeah, I've seen a lot of pancreatic cancer, just very, very difficult to treat.
Yeah.
Yeah. I guess for me, I mean, there's obviously importance with market and challenges and adoption and all that, but when you think about personalized medicine, what gets you the most excited?
It's the goal that we're trying to achieve, the needle-to-needle time, helping patients, critical patients, and it's just a revolutionary technology, and getting there. I mean, being part of that is just phenomenal. And I know the team that we have back at our facility in Torrance, where our NAP lab is established, are really excited and proud of what they do, and why they're doing it. There's not many labs in the world that do this, and we're one of the few, and we have a GMP site that does this. So it's just incredible to be part of that.
Yeah, definitely. How about you?
I think everybody's been touched by cancer and knows people in their lives, and the typical treatments, they're really hard on people, radiation, chemotherapy, and it's just the sadness of, everybody's cancer is different, and so, it's unpredictable what helps somebody doesn't, and I think this technology offers the potential to not leave anybody behind, and a much easier type of therapy for people, and using your body's natural immune system. I mean, it's really powerful, and so I feel like I want to, as a company, support it any way we can, because I think it really offers the potential to help save many people's lives.
Right. Yeah, I do think that it being this new area, it does give us an opportunity to be innovative.
Yeah.
So I think there's a lot of exciting things that will come out because of it, like just on the science and technology side, right? Even just beyond the neoantigens, like new things will emerge because we have to address these specific concerns.
Absolutely. Yeah. And that's science. That's what happens.
Yeah, that's right. Absolutely. That's science.
Yeah.
All right. Great. Well.
And I just want to say too, Trishul, it's been a real pleasure working with you over the years, and
you too.
And PolyPeptide's a great company to work with and very innovative. And
yeah, we appreciate all the support that CM has given us in this area. Been a good partner to us.
Absolutely.
We can't do this without our partners, and CM is vital and important to that.
Definitely. We've learned a lot working with PolyPeptide, and you've really helped us to make our equipment a lot better.
Yep.
So thank you.
Absolutely. Thank you.
All right. Well, thank you both for your time. I'm really excited for the rest of the conference, of course. We're here at TIDES 2021 in Boston, so excited to see some of the lectures and hang out at the booth. And again, thank you for taking the time to walk us through the process, to talk about your thoughts and feelings on it. And we look forward to collaborating with you more.
Absolutely.
Thank you for tuning in to Exploration Science. We welcome your feedback on this topic, as well as suggestions for topics that you'd like to see covered. You can also find this content available as a blog post and a podcast using the links in the description. Thanks again for tuning in.